Hi everyone, my name is Juvissan and today I will tell you a little bit about myself and my life as an early stage researcher.
I am original from Peru, but I lived in Brazil the last 6 years. In Peru, I got my bachelor's degree in pharmacy and biochemistry at San Marcos University. Then I got my master's degree in biochemistry at Federal University of Rio de Janeiro. I always wanted to study and have the experience of living in Europe, so I applied for PhD positions. Currently, I'm doing my PhD in biotechnology and working as an early-stage researcher for synBIOcarb. synBIOcar is a Marie Curie program which enables universities, research centers, and companies across Europe to host talented foreigner researchers by providing the necessary skills and international experience. We are 15 PhD students in synBIOcar network, and all the projects are focused in the field of glycobiology. Basically, we are using different approaches, but at the same time, it allows us to collaborate between us. Okay, let's start from the beginning. As I mentioned, I am based in Slovakia, and I will show you how is my daily life at the Slovak Academy of Science. I am working at the Institute of Chemistry under the supervision of Dr. Jan Kach, and our group is focused in lectin-based affinity biosensors using nanotechnology. In my project, my role is to identify and characterize overall glycans. They are gaining interest in biomarker discovery and can be used as promising targets for the development of biosensors in early diagnosis of several cancers. Biomarkers are molecules which provide useful information about the physiological state or change during a disease process, and the good point is that they are measurable, so it's possible to observe some changes in the DNA, proteins, or glycans. At Institute of Chemistry, we are using cutting-edge technology for identifying specific glycans. But why glycans? Glycans are complex carbohydrates attached to proteins or lipids. Approximately from 50 to 80% of the human proteins are glycosylated. Glycans play a critical role in biology, like how the body recognizes and fights with viruses like SARS-CoV-2, which is highly glycosylated and allows to shield from the immune system. But what is more important for us is that some studies have shown that cancer cells release glycans at the early stage of cancer, and the changes in the glycosylation pathways are associated with cancer progression. But why is it challenging to study glycans? Because of the lower amounts and diversity of glycoforms and microheterogeneity, and for this we need very sensitive and high throughput techniques, and this is the first objective of my project. So in order to have with confidence and go further with the biomarker candidate, we really need some platforms. In our lab we are using mass spectrometry to screen for looking for a potential glycan target. I think MS is a major tool for characterizing glycans, and I really enjoy working with this technique. Now we have to discuss another important issue, that is the clinical performance of biomarkers used to diagnosis of colorectal cancer, for example. They have a poor specificity and sensitivity, and only the 40% of patients with CRC are diagnosed at early stages. The common tests are colonoscopy, proctoscopy, and biopsies. Unfortunately, most of these exams are expensive, invasive, and uncomfortable for patients. And this is the second objective of my project, to develop a bioanalytical device or biosensor with enhanced specificity and sensitivity. Basically, a biosensor can transform information from a specific sample into an analytical signal. In this diagram, we can see that a biosensor usually contains three main parts. A biorecognition element can be lectin or antibody, which will interact specifically with the analyte, and a physicochemical transducer, which converts the physicochemical signal of biorecognition into an electrical or optical signal that will be proportional to the analyte concentration. And this is why we are gonna use optical methods such as surface plasmon resonance and electrochemical methods. Our plan is to couple with lectins, glycoproteins, antibodies, and nanomaterials and construct glycan binding modules to develop new biosensors, which would have the potential to detect changes in the glycan composition with more specificity and sensitivity. Here we can see that there are various possible biosensor configurations: lectin carbohydrate, carbohydrate lectins, and antibody carbohydrate lectin that can be immobilized on surface of electrodes. With the new advances in nanotechnology, sensors based on nanomaterials, nanocomposites have become important tools as an alternative to expensive and complex devices for health monitoring systems. They can provide very low detection limits with range from picomole to attomole. In my project, I will use MXins, since has been pointed as a material of the future in the new field of two-dimensional nanomaterials because of their exceptional properties such as good electrical conductivity, high surface area, hydrophilicity, electrochemical stability, biocompatibility, and easy of surface functionalization. Now I will show you how to construct a biosensor. First, we have to pre-treat our bare electrode. Then, we will modify it with the dispersion of MXin. Also, we can add a monolayer with carboxylic groups. At this point, we can immobilize it with antibodies on surface via amine couple through ADC-NHase chemistry. After that, we can use a blocking agent, and our biosensor is ready to recognize the target antigen. Finally, the resulting current is measured. We will get a Nyquist plot, where a dynamic of an electrochemical system can be observed. The semicircles show the charge transfer resistance after every building step of the biosensor. What is very awesome from this program is that provide the financial support to have advanced training courses, and we can participate of international meetings and conferences. It was awesome to visit Graz, Austria, where I did my secondment at ASIP. Bratislava is a lovely place, quiet, and what I really like is that I am really close to nature. Slovakia matched very well with my personality. So, thanks for watching, and if you are interested in expanding your knowledge in glycobiology, you can see what others ESRs are working on. Please don't forget to visit synBIOcarb portal and follow us in Twitter.